Hello and good morning. Today we're going to delve into Chapter 7, Mutation. So this is the chapter that you should have been exposed to when you entered the lecture slot last week, where you answered the Google form. So just an overview of what this chapter will be like. Chapter 7 is divided into 3 subtopics. So the first one is mutation classification and types, where you will define mutation, you will state the 2 types of classification, and you will state the 2 types of mutation, and also what causes them. Secondly, you will learn about gene mutation, also called point mutations, and this will be subtopic 7.2. And subtopic 7.3 is chromosomal mutation. These are the learning outcomes that we expect you to be able to understand. And now let us talk a little bit more about mutation itself. So we can define mutation as a permanent change in the genetic information or the DNA of an organism. So as long as your DNA has changed, even just a little bit, that is already a mutation. That mutation can occur in your somatic cells, for example, in your skin, in your bones, and even in your liver. And if it occurs in your somatic cells, then it will stay in you. But if it occurs in your gametic cells, which is in your eggs and your sperm, it can be passed on to the next generation. So any organism that is carrying a mutated gene or any gene that has undergone a mutation is called a mutant. So these are examples of mutants. These are mutant vegetables and mushroom. Some of them have strange shape that doesn't even look like a broccoli anymore. And they are examples of mutant animals. So this is an example of a mutant Drosophila melanogaster. A normal Drosophila is supposed to look like this with 1 pair of wings. But this mutant has 2 pairs of wings. That is because there is a mutation in its bithorax complex. So the gene that is controlling how many wings the Drosophila has been mutated, and so it has an extra pair. So there can be mutant bacteria, mutant virus, mutant plants, mutant animals, and mutant humans. Plants, animals, bacteria, virus, if you want to call them mutants, that is perfectly fine. But generally, it is not acceptable for you to call people mutants, because although people who carry mutated genes, in terms of science, they are organisms that carry a mutated gene, it's still not very nice for you to look at someone with a disease and then call them a mutant. Unless you are watching X-Men or you're watching any superhero show, please don't call humans mutants. Okay? Right. Now we are going to go into the 2 types and the 2 classifications of mutation. So you have gene mutation, which occurs in only the gene. So if this is your chromosome, it affects only the gene. But chromosomal mutation affects the entire chromosome. So those are the 2 classes of mutation. What are the 2 types of mutation? Mutation can occur spontaneously. So that's spontaneous mutation. Or it can occur because it is being induced by something outside. So if it is spontaneous mutation, it can occur during DNA replication. During DNA replication, mistakes occur and the DNA just comes out wrong. Or it can occur because of non-disjunction during meiosis. So this is an example of that mutation that occurs during DNA replication. And this is an example of the mutation that occurs during non-disjunction in meiosis. So you start with the diploid, haploid, 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 haploid daughter cells. if you have non-disjunction during the second meiosis, this is a bit of an error on the slide, please make the correction. You start with a diploid, you end up with haploid. But because of non-disjunction, this blue one, the sister chromatids do not separate properly during meiosis 2. And so you end up with N, missing one chromosome, N, extra one chromosome, normal haploid, normal haploid. In the abnormal, you start with a diploid, non-disjunction occurred during meiosis 1, you end up with haploid, missing one, haploid, extra one. You had a normal meiosis 2, and you end up with haploid, missing one, haploid, missing one, haploid, extra one, haploid, extra one. Okay. And then we talk about induced mutations. Induced mutations, it can occur because of mutagens. Mutagens are agents that cause mutation. So these agents that cause mutation can be physical mutagens or chemical mutagens. Physical mutagens, their examples are ultraviolet rays, gamma rays, X-rays. So this is an example of that UV ray on your DNA. So when the UV rays are when the DNA is exposed to UV rays, the thymine bases can sometimes form thymine dimer. That means instead of holding hands with the base that is across from it, it holds hands with the thymine next to it. What happens is, when this thymine dimer forms, the DNA polymerase cannot read this sequence of DNA properly, and so the DNA replication will go wrong. Then this is an example of colchicine effect on chemical mutagen effects. So the colchicine's effect is it will prevent the formation of microtubules in cells during mitosis and meiosis. So what happens is you end up with polyploidy. The cells, instead of being haploid or diploid, they may end up as tetraploid, which is 4N. So we can use these mutations to our advantage. In this example, we can use colchicine to produce tetraploid plants, which have bigger leaves, bigger fruits, and therefore give us a higher yield. Okay, hasil kita lebih banyak. And we can also use it in the medical industry. We can use it to prevent tumor cells from dividing in certain chemotherapy treatments. Alright, so that is it for now. Let's take a short break before we go into gene mutation.